The idea that the animal kingdom is so diverse, so widespread, and we're going to classify why it is in that orientation and in that pattern. Now, one of the historical components that we have to understand is the following. The Cambrian Explosion is a big reason for the great amount of animal diversity on planet Earth. The Cambrian Explosion happened about 535 to 525 million years ago. So I'll just write this down on the top as a side note. 535 to 525 million years ago, MYA, just as a point of reference. Now, the reason why we mention this in a Gen Bio class is because this is going to be a point at which we have the rapid, that's why it's called an explosion, Cambrian explosion, a rapid appearance of many different animal body plans. Rapid appearance of many diverse, many different animal body plans. Animal body plans. And we established that body plans are a good point of comparison, a good point to look at key evolutionary features because of their unique developmental patterns and unique developmental overall results based off of what the body plan actually is. And thus, at this time, in this time period, we have a huge explosion of different body plans. And in addition, in the Cambrian explosion, this is when we actually also see the first fossils ever, of large animals. So this is the first time we see animals getting bigger. We see animals getting more and more successful. And these large animals have hard mineralized skeletons with hard mineralized skeletons. What does that tell you as an evolutionary biologist, let's say? It tells you that during the Cambrian explosion, the animals that were developing here not only had the many different body plans, but were large. That would mean that they actually had the capability of getting enough food, of getting enough energy, of getting enough success in their environment and in their world to grow to greater sizes than ever seen before. Thus, this is the first moment in which we have the typical animals that we think of today: cat, dog, mouse, whatever it may be. All of those are typically considered large animals as opposed to smaller subcellular or microscopic type of animals. So that's our Cambrian explosion idea. That's going to play a big role in our diversification. And then in addition, another sort of side thing we have to establish is that most of the current phyla of animals, most of our current phyla of animals, meaning that the current groups of animals all throughout the world, that's what phyla means. Most of current phyla of animals were actually established 500 million years ago. So basically, right after the Cambrian explosion, after this large diversification event, we're going to have most of everything that we see on Earth today. All of the animals that we see today have themselves established about 500 million years ago. This would be about, in total, about 36 different phyla. Phyla are huge, large classes of organisms, and we have 36 different phyla of animals that are extinct. That would mean extant means that they are living today, as basically the opposite of extinct. When you have an extant species, it's a living species. And this all gives rise to something that we haven't talked about yet, but the idea of kingdom Animalia. Kingdom Animalia has about 1.3 million species within it, and we've seen this number before, and you obviously know what I'm going to compare this with. Not only does it have about 1.3 million identified species, but scientists taxonomists estimate that there are 10 to 20 million species out there that have not been totally recognized or established in terms of classification. Estimated 10 to 20 million species. So we're not even halfway there in terms of how many we've discovered. So that's our historical component to understand about the diversification. Now, an important part of diversification is animal phylogeny. Animal evolutionary history, in other words. And we've touched upon many different phylogenetic moments of animal history as we've gone through this lecture. Now we're going to just classify them a lot more formally. And that's going to be done by especially looking at figure 32.11 and also by looking at the many different key animal features that we've looked at throughout this lecture. Now, part of the phylogeny that we have to understand, the evolutionary history, is a very big first important fact of animals. All animals, and this is why a lot of phylogenetists would like to study animals, and that's why they're a good point of study, because all animals share a single protist common ancestor. A single protist common ancestor. So, if we have animal phylogeny, if we have an animal history, the great thing about studying this is that we have a route at which we can explore everything that comes from this one single protist common ancestor. That single protist common ancestor was the choanoflagellate, and we've briefly mentioned it when we talked about the idea of uniconts at the end of our protist lecture. But this is a big important fact because this tells us that kingdom Animalia, everything that is an animal on planet Earth, is part of a large monophyletic group. And being monophyletic, as we know in terms of phylogeny, is great to have one common ancestor. You're not paraphyletic, you're not polyphyletic, you're monophyletic. One root to establish everything from. And that's where the power of the animal phylogeny comes from, and the power of studying it also comes from. So, within this monophyletic group, what we're going to first establish is the following. We have within kingdom Animalia is the idea of the first evolutionary trait that we actually looked at, and key animal feature was the idea of true tissues or no true tissues. And that can be classified as whether or not you are a metazoa, so the metazoa clade, versus over here the eumetazoa clade. So if you are of the metazoa clade, there's only one option that you need to know of. That means that you are probably going to be a sponge. These are sponges. And sponges are equal to basal animals, which means they are the base of the entire flowchart. The base of the entire animal world are sponges because they were the first. Okay? They are branches that form the base of the tree. Periphery, in other words, are branches that form base of our animal phylogeny tree. And this can be seen if you look at figure 32.11, much clearer than as depicted here. And because of this, we also are going to just remember that this will classify further into the phylum Periphery if we want to be specific. And we talked about Periphery before. Periphery, thus, are definitely going to be monophyletic organisms, monophyletic, because they come from a single protist common ancestor known as the choanoflagellate. So that's our metazoa clade. Now, very simple organisms. They're the exception to every animal rule. What about the more complex eumetazoans, those that have true animal tissues? Well, these are going to be, and we're going to write this down, as true tissue organisms. And when you are a true tissue organism, you have the option of symmetry, right? Here we have asymmetry, if you remember. Over here, we're going to have two types of symmetry, and that is either radial symmetry or bilateral symmetry. Now, the first one that we're going to go over is the idea of the Clade Radiata. And that would mean somebody that has radial symmetry versus the Clade Bilateria, which is somebody that has bilateral symmetry. So this is our first moment of divergence between the eumetazoans. What type of symmetry do you have? In Clade Radiata, we're actually going to establish now two specific phyla that you have to remember that are radially symmetric. And those are the phyla Ctenophora, Ctenophora. This is not pronounced Ctenophora or Ctenophora. It's actually a silent C. It's a Greek word. Phyla Ctenophora. And then we also have plus phyla, same idea here, silent C, Cnidaria. You have to remember these two, and you have to remember that they're part of the Radiata clade, and you have to remember that they are eumetazoans. And you have to remember that they are grounded on this single protist common ancestor. So what are these two things? These two things are going to be broadly referred to as basal eumetazoans. So not just basal animals here, they're actually specifically a type of basal animal, specifically basal eumetazoans. So let me write this down as metazoans. So they are the first eumetazoans, the first to have true tissue. They are diploblasts. So reason why they're diploblasts is because they have this radial symmetry, right? We know bilateria must be triploblast, and we'll get to that. Diploblasts, and they of course have radial symmetry if it's not already obvious from their name. And we'll just write this down. So those are 3 characteristics you have to remember about Ctenophora and Cnidaria. Now, off to bilateria, where we have even more complexity now. Over here, bilateria, what you need to remember is that this is actually going to be most animals. Most animals are of the clade bilateria, and remember, clade bilateria is a subclade of the eumetazoan clade, of those who have true tissue. Their symmetry is bilateral. So most animals are here. They have bilateral symmetry. We'll write this down. These are 3 characteristics that you need to remember. Most animals, bilateral symmetry and bilateria, and also, of course, triploblasts. All bilaterals are triploblasts. So 3 things: most animals, bilateral symmetry, and triploblasts. Now, we can actually name some of these organisms, and they are specifically going to be either protostomes or deuterostomes. This is where we have that divide. Remember, we talked about this. And now there are specific organisms you need to know. So let's do protostomes here. It's actually going to be called now not just protostomes, but we'll be more specific in our classification. It's called Protostomia, the larger family, and then over here we're going to be doing Deuterostomia. And we know what this means, of course: second mouth, first mouth. In Protostomia, the 2 types of organisms that you need to know are known as the Lophotrochozoans, and let me spell this correctly, hopefully. Lophotrochozoans. And in the other side, there are also going to be the ecdysozoans. Ecdysozoans. So, what do these things have? What are their characteristics? Lophotrochozoans are only going to be invertebrates. We'll get to the idea of vertebrates in just a second. Inverts would just stand for invertebrates. They have a lophophore feeding structure. Your textbook has a good figure of this. They have a lophophore feeding structure, thus the name, Lophotrochozoans. And one last thing to remember about these guys, they also have a trochophore larval stage, thus the name Lophotrochozoans. Trochophore larval stage, and we know all about larval stage versus non-larval. And on the ecdysozoan side of this story, look at figure, we can do this underneath here, for Lophotrochozoans, look at figure 32.12, just to get a better idea of what this means. And on the ecdysozoan side, these are also going to be only invertebrates, so inverts for invertebrates, and they will undergo a process known as ecdysis. That's the name. Ecdysozoans undergo ecdysis. Ecdysis is just another word, a fancier word for the process of molting, of shedding a shell, essentially. And molting would be an exoskeleton or cuticle. So they possess an exoskeleton or something known as a cuticle that they have to shed off. Thus, they undergo the process known as ecdysis to shed off that exoskeleton or cuticle structure. So that's protostomia, and then finally, last part is deuterostomia. Now, in deuterostomia, in this class, we're going to have the phylum to remember is Chordata, and we're going to talk about this in much greater detail in the next lecture, actually, but we're just introducing it here. Phylum Chordata. This will include the vertebrates, but also some invertebrates, so it's not completely grounded on only vertebrates and only invertebrates. There's actually both in this situation. And also, deuterostomia actually does not contain, they're actually not all, specifically, not all completely deuterostomes. Okay? They're not all deuteros. And we'll get into those details in our next lecture.